Hello, friend, and welcome to Facts for Sleep. Tonight, we're going to explore something that most people never think about. We're going to look at the world through a very different kind of eye. The eyes of insects. If you find this kind of slow exploration helpful, you may want to subscribe. There is no rush here. Just a quiet journey through the science of how tiny creatures perceive the world around them. Most of us move through life trusting our own vision completely. We assume that what we see is simply what is there. But vision is not a passive recording of reality, it is a construction. The brain takes raw signals from the eye and builds an image. Different animals build very different images from the same physical world, and among the most different of all are the insects. Their eyes are built on a completely different design. They evolved along a separate path, and the images they form are unlike anything a human brain would recognize. To understand insect vision, we have to start with the eye itself. The human eye is a single lens eye. There is one lens at the front. It focuses light onto a sheet of receptor cells at the back. That sheet is called the retina. The retina contains millions of cells that are sensitive to light, and these cells send signals to the brain. The brain then assembles those signals into a continuous image. This design is called a camera eye. It produces sharp, detailed images. It is very effective for seeing fine detail at a distance. Insects do not have camera eyes. Most insects have what are called compound eyes. A compound eye is not a single lens focused on a single surface. It is a collection of many small individual units. Each unit is called an ommatidium. Every ommatidium is a tiny optical structure. It has its own lens at the top. It has its own set of light-sensitive cells below, and it collects light from one small portion of the visual field. Together, the hundreds or thousands of ommatidia in a single compound eye each point in slightly different directions. They cover a wide area of space. Each one contributes a small piece of information. The brain assembles all those pieces together. The word ommatidium comes from the Greek word for a eye. The plural is ommatidia. A single compound eye in a dragonfly can contain as many as 30,000 ommatidia. A housefly has around 4,000 in each eye. A honeybee has around 5,000 in each eye. The number of ommatidia varies greatly between species. It depends on the lifestyle of the insect and how much visual detail it needs. A cave-dwelling beetle that rarely sees light may have only a few hundred ommatidia, or may have reduced eyes entirely. A predatory wasp that hunts in bright daylight may have large, densely packed compound eyes that give it the best possible visual resolution within the limits of this design. Each ommatidium is shaped somewhat like a long, narrow tube. At the very top there is a transparent lens called a corneal lens. Just below that is a second lens called the crystalline cone. These two lenses work together to focus light down into the tube. Below the crystalline cone is the rhabdom. The rhabdom is a structure made from the light-sensitive parts of the receptor cells. These receptor cells are called retinular cells. In most insects, there are between 6 and 9 retinular cells arranged around the central rhabdom. The rhabdom acts as a light guide. Light that enters the ommatidium travels down through the rhabdom. The retinular cells detect the light and send signals upward into the brain. The angle of acceptance of each ommatidium is quite narrow. Each one captures light from only a small cone of space in front of it. In many insects, this cone is roughly 2 to 3 degrees wide. Because the ommatidia point in different directions, together they cover a very wide visual field. But because each one captures only a tiny slice of that field, The overall resolution of the compound eye is much lower than the resolution of a human eye. The compound eye trades sharpness for coverage. To understand what this means in practice, consider the following. A human retina contains roughly 120 million rod cells and 6 million cone cells. These provide extremely fine resolution. An insect eye has far fewer individual light-sensing units. Even a dragonfly with 30,000 ommatidia has far less resolve and power than a human eye. If you could look at a printed page through a dragonfly's compound eye, you would not be able to read the letters. The image would be coarse and pixelated like a very low-resolution photograph. Instead of a clear image, there would be a mosaic of light and dark patches. The outline of large objects would be visible. Broad regions of colour or contrast would be apparent. But fine detail would be lost. But this is only part of the story. Because the low resolution of the insect eye does not mean insect vision is inferior. It means insect vision is different. The compound eye excels at things the human eye is not as well suited for. And understanding those things helps us see why this eye design has survived for hundreds of millions of years. The compound eye first appears in the fossil record in the early Cambrian period. That was roughly 530 million years ago. Some of the oldest known animal eyes are compound eyes. They have been found in the fossils of trilobites. Trilobites were arthropods, distant relatives of modern insects and crustaceans. Their compound eyes were already sophisticated structures. Some trilobite species had calcite lenses in their ommatidia. Calcite is a crystalline mineral. Using calcite as a lens material is unusual. In most modern compound eyes, the lenses are made of transparent biological material. But calcite has optical properties that make it well suited for forming images underwater, where trilobites lived. The fact that trilobites evolved such specialized lenses tells us something important. Vision was already a strong evolutionary priority in the Cambrian. Animals that could see well outcompeted animals that could not. The compound disc in various forms has proved enormously successful. Today it is found not only in insects but also in crabs, shrimps, lobsters and many other arthropods. One of the most important advantages of the compound eye is its wide field of view. Because the ommatidia point in many different directions, a compound eye can see a very wide angle of space at once. In many insects, the compound eyes are large and curved, positioned on the sides of the head. This gives them the ability to see in almost all directions simultaneously. A dragonfly, for example, can see nearly 360 degrees around itself. It has very little blind spot. A honeybee can see forward, sideways, and partially behind itself at the same time. This wide-angle vision is very useful for detecting approaching predators or tracking fast-moving prey. A second important advantage is sensitivity to motion. The compound eye is exceptionally good at detecting movement. This is because any moving object crosses the visual fields of multiple ommatidia in rapid succession. As the object moves, different ommatidia are activated one after another. The insect brain is very sensitive to these rapid changes in activation. It can detect movement much faster than a human brain can. This is why it is so difficult to swat a fly. The fly detects your approaching hand through motion detection long before the hand arrives. By the time your hand reaches the spot where the fly was, it has already reacted and moved away. The speed of insect motion detection is linked to a property called the flicker fusion rate. The flicker fusion rate is the speed at which a series of rapidly changing images begins to look like a smooth, continuous image, rather than a sequence of separate frames. For humans, this rate is roughly 24 to 60 frames per second, depending on conditions. This is why films recorded at 24 frames per second appear as continuous motion rather than slideshow. For many insects, the flicker fusion rate is much higher. A housefly has a flicker fusion rate of around 300 frames per second. This means the fly perceives the world as a series of distinct moments that are separated in time far more finely than we are capable of seeing. To a fly, a film playing at 24 frames per second would look like a rapid slideshow of still images, not smooth motion. The world appears to move in much finer time steps from the fly's perspective. This high temporal resolution is enormously useful for a fast-flying insect navigating through complex environments. A fly travelling at speed needs to detect obstacles and make evasive manoeuvres in fractions of a second. The faster the visual system can update its information about the world, the more accurately and safely the insect can move. Over time, the compound eye and the fast-processing insect brain evolve together as a matched system. The eye produces rapid updates. The brain processes them quickly. The body responds in time. Now we move to something that changes the picture even more dramatically. It concerns not the speed of insect vision, but the range of colours they can see. Human vision covers a range of the electromagnetic spectrum from roughly 380 nanometers at the violet end to roughly 700 nanometers at the red end. This range is called the visible spectrum for humans. Within this range, we can distinguish millions of different shades of colour. We do this because our retinas contain 3 types of colour-sensitive cells. These are called cone cells. One type is most sensitive to short wavelengths, which we perceive as blue. One type is most sensitive to medium wavelengths, which we perceive as green. And one type is most sensitive to long wavelengths, which we perceive as red. By comparing the signals from these 3 types of cones, the brain can reconstruct colour. Insects do not all use the same 3 colour channels. Many insects have photoreceptors that are sensitive to different parts of the spectrum than human photoreceptors. Crucially, many insects are sensitive to ultraviolet light. Ultraviolet light has wavelengths shorter than the human visible range. It falls below 380 nanometers. Human eyes cannot detect ultraviolet light at all, but the photoreceptors of many bees, butterflies and other insects are tuned to detect it. The honeybee is one of the most studied examples. The honeybee has 3 types of photoreceptors. One is sensitive to ultraviolet light, one is sensitive to blue light, and one is sensitive to green light. Notice that honeybees have no red-sensitive photoreceptors. This means that a red flower looks very dark to a honeybee. It reflects very little ultraviolet, blue or green light. The bee does not perceive it as bright or vivid. However, many flowers that look plain white or pale yellow to human eyes appear strikingly patterned in ultraviolet to a bee. This is because many flowers have ultraviolet absorbing pigments in their petals. These pigments create patterns that are invisible to humans but clearly visible to bees. These hidden patterns are called nectar guides. They are markings that guide pollinators toward the center of a flower where the nectar and pollen are found. When photographed with cameras sensitive to ultraviolet light, many ordinary-looking flowers reveal vivid patterns. A pale yellow buttercup, for example, reveals a dark central zone surrounded by a brighter outer ring. The center of the flower absorbs ultraviolet while the outer petals reflect it. To a bee, this creates a clear visual target pointing directly to the reward. Sunflowers display particularly striking ultraviolet patterns. What appears to a human as a uniformly yellow ring of petals shows up under ultraviolet photography as a two-toned arrangement. The outer portions of the petals reflect strongly while the inner portions absorb, creating a broad arrow pointing inward. The visual signal is precise and unambiguous. The relationship between insect ultraviolet vision and flower markings evolved together over a very long period of time. Flowering plants that developed ultraviolet markings attracted more pollinators. They were therefore more successful at reproducing. Over millions of generations, flowers evolved increasingly elaborate ultraviolet patterns. At the same time, insects that could detect these patterns were more efficient at finding food. The two evolutionary pressures reinforced each other. The result is a visual communication system between insects and plants that is entirely invisible to our eyes unless we use special equipment. Butterflies extend this story even further. Many butterfly species have 4 or even 5 types of photoreceptors in their eyes. Some species can see well into the ultraviolet. They can also perceive polarized light and have receptors sensitive to wavelengths in parts of the spectrum that human scientists are still working to fully characterize. The swallowtail butterfly, for example, has been found to have at least 5 distinct types of photoreceptor. With 5 color channels instead of 3, the swallowtail can theoretically distinguish an enormous number of color combinations that would be indistinguishable to a human observer. 2 objects that look identical in color to our eyes might appear completely different to a swallowtail. Male and female butterflies of some species look nearly identical to human eyes. But when photographed in ultraviolet light, clear differences between sexes become apparent. In these species, the ultraviolet pattern is thought to be the primary signal used in mate choice. Females may assess the quality and brightness of a male's ultraviolet pattern as an indicator of his health and genetic quality. Because ultraviolet reflectance depends on the precise nanostructure of the wing scales, it is a signal that is difficult to fake. Only a genuinely healthy butterfly can maintain the elaborate scale architecture needed for high ultraviolet reflectance. The wing scales of butterflies are among the most structurally complex biological surfaces known. Each scale is a thin, flattened plate attached to the wing membrane at one end. The surface of the scale is covered with an intricate arrangement of ridges, ribs, and crosslinks made of a transparent protein called chitin. The spacing between these nanostructures is on the order of hundreds of nanometers. When light hits these structures, it interferes with itself in a phenomenon called structural coloration. Rather than absorbing some wavelengths and reflecting others through pigment chemistry, the nanostructure of the scale physically filters wavelengths based on their interference with the periodic structure. The colors produced by structural coloration are often iridescent. They change with viewing angle. The brilliant blue of the morpho butterfly is not produced by any blue pigment. It is entirely the product of structural coloration. And many of these nanostructures scatter and reflect ultraviolet wavelengths with particular efficiency, creating the ultraviolet patterns that are so important for butterfly communication. Dragonflies are even more remarkable in terms of their photoreceptor complement. Research has shown that some dragonfly species may have as many as 11 or even 30 different types of photoreceptors. This is an extraordinary number. For comparison, mantis shrimps, which are crustaceans rather than insects, hold the record among animals with 16 types of color receptors. The dragonfly approaches this with its own impressive array. However, having many photoreceptor types does not necessarily mean superior color discrimination in the way humans understand it. Recent research suggests that dragonflies may use this wide spectral coverage differently. Rather than comparing signals across many channels in a sophisticated way, they may use a simpler system that rapidly matches incoming light to known patterns. This would make them fast and effective rather than analytically precise. Among the most visually complex of all insects are the mantids. Mantis species have highly developed front-facing eyes that give them good binocular vision in a narrow forward zone. Binocular vision occurs when both eyes can see the same point in space simultaneously. When this happens, the brain can compare these slightly different views from each eye and use the difference to calculate depth and distance. This is called stereoscopic vision. Humans rely heavily on stereoscopic vision for depth perception. Mantids also use it, but in a very different way. Research published in recent years has shown that the praying mantis uses a unique form of stereoscopic vision that works at the temporal level rather than the spatial level. Instead of comparing two simultaneous images from different positions in space, the mantis compares images from two slightly different moments in time. This gives it depth perception that works particularly well for moving targets. It helps the mantis judge the distance to a moving prey animal with great precision just before striking. We should also consider what insects do not see, as well as what they do see. Most insects have limited ability to focus at different distances. The lenses of Ommatidia are fixed. They cannot adjust their focus the way the human eye can. As a result, insects generally have a fixed focal distance. Objects at that distance are as clear as the compound Ialum. Objects at other distances become blurred. The range at which compound eyes work best is relatively short. Most insects have their best vision within a few centimeters to a few meters. At very long distances, the resolution of the compound eye is simply too low to be useful for detailed object recognition. This limitation is partly compensated for by behavior. Many insects will move their heads or bodies to scan the environment from slightly different angles. This generates a kind of motion parallax, where nearby objects appear to shift more than distant objects as the viewing position changes. The insect brain uses this shift information to estimate distance. Locusts are well known for performing characteristic peering movements, swaying their heads from side to side before jumping. This behavior is thought to help them judge the distance to a landing spot. The size of the image shift produced by the peering movement depends on the distance to the object. Nearby objects shift a great deal. Distant objects shift very little. By measuring this shift, the locust extracts distance information from the motion of its own head. This is a clever solution to the problem of depth perception in an eye that lacks the ability to focus. Some insects also have additional simple eyes called ocelli. Ocelli are different from compound eyes. They are small, lens-covered photoreceptors that are usually found in a triangular arrangement on the top of the head between the compound eyes. Most flying insects, including bees, wasps, dragonflies, and many others, have 3 ocelli. These are not used for forming images. They are not capable of that level of detail. Instead, the ocelli function as rapid horizon detectors. They are very sensitive to changes in light intensity across a wide visual field. They help insects maintain its orientation relative to the horizon and detect rapid changes in illumination, such as the shadow of a predator. Because the ocelli connect directly to fast motor pathways in the nervous system, they can trigger stabilizing reflexes in the flight muscles with extraordinary speed, faster than the compound eyes can relay information. The lenses of the ocelli are actually too large for the eye to focus a sharp image onto the underlying receptor cells. The focal point of the lens falls behind the retina of the ocellus. This might seem like a design flaw, but it may actually be a useful feature. Because the image is deliberately defocused, each receptor cell in the ocellus receives light from a broad area of space. This makes the ocellus extremely sensitive to overall light level. It responds strongly to any change in the overall illumination of its visual field. It is optimized for detecting large, fast changes in brightness, rather than for resolving fine spatial detail. An approaching predator creates a sudden shadow that the ocellus detects immediately and reports to the flight control system in a fraction of a millisecond. The design of compound eyes also varies considerably between species depending on lifestyle. Insects that are active in low light conditions, such as moths and many beetles, often have what are called superposition compound eyes. In the more common type of compound eye, called the apposition compound eye, each ommatidium is optically isolated from its neighbours. A narrow absorbing layer between the tubes prevents light from one ommatidium from leaking into adjacent ones. This ensures that each unit only responds to light coming from its own narrow field of view. In superposition eyes, this isolation is removed or reduced. Light from many ommatidia is combined and focused onto a smaller number of receptor cells. This dramatically increases the sensitivity of the eye in dim conditions. It allows the insect to see in near darkness that would be effectively invisible to an apposition eye. The trade-off is reduced resolution. Superposition eyes produce a brighter but less detailed image. Some moths that fly at dusk and dawn have superposition eyes that are so sensitive they can navigate by the light of stars. This is a remarkable ability. It requires not only a very sensitive eye, but also a brain that can interpret the dim and scattered light field of a starlit sky as a meaningful navigational signal. Moths and other nocturnal insects use the positions of celestial objects to maintain a straight flight path. They keep a fixed angle between their body axis and the light source. When the light source is very far away, as stars are, this strategy produces a straight line of travel. When the light source is nearby, as a candle or a street light is, the same strategy produces a spiral path inward toward the source. This is why moths appear to spiral toward artificial lights. It is not that they are attracted to heat or light in a simple way. It is that their ancient navigational strategy, designed for celestial bodies at effectively infinite distance, is being applied incorrectly to a nearby light source. The polarization of light is another dimension of visual information that many insects can detect. Polarization refers to the direction in which the electric field component of light waves oscillates. Sunlight is unpolarized when it leaves the sun, but when it scatters through the atmosphere or reflects off surfaces such as water, it becomes partially or fully polarized. Insect eyes, particularly the ommatidia and certain regions of the compound eye, contain photoreceptors that are sensitive to the plane of polarization. This gives insects access to visual information that is completely invisible to unassisted human eyes. The rhabdom of each retinular cell contains a large number of tiny parallel structures called microvilli. These microvilli are tubular extensions of the cell membrane. The light-absorbing protein molecules called rhodopsins are embedded in the membranes of the microvilli. Because the microvilli are aligned in a regular direction within each retinular cell, the rhodopsin molecules within them are also aligned. This means that each retinular cell preferentially absorbs light, whose polarization plane is parallel to the orientation of its microvilli. Different retinular cells within a single ommatidium have microvilli oriented in different directions. This gives the ommatidium sensitivity to multiple planes of polarization simultaneously. By comparing the signals from cells with different microvilli orientations, the insect brain can determine the polarization angle of incoming light. Honeybees use polarized light as a navigational tool. The sky, even on a partly cloudy day, contains a predictable pattern of polarization based on the position of the sun. Even when the sun itself is behind clouds, the pattern of polarized light in the visible portion of the sky allows the bee to calculate where the sun must be. This lets the bee navigate by the sun even when direct sunlight is unavailable. The bee's polarization-sensitive photoreceptors are concentrated in a special dorsal region of the compound eye known as the dorsal rim area. This region points upward and backward toward the sky. It is dedicated specifically to reading polarization information from the sky dome. Water surfaces also reflect polarized light. Many insects, including mayflies and dragonflies, use polarization detection to locate water. The light reflected from a smooth water surface is strongly horizontally polarized. This creates a distinctive polarization signal that a polarization-sensitive insect can detect from a distance. Mayflies, for example, lay their eggs on water. They need to find suitable water bodies as part of their reproductive cycle. Research has shown that polarized light from water acts as a strong attractant signal for mayflies. Unfortunately, this sensitivity has a dark side in the modern world. Many artificial surfaces, including cars, solar panels and asphalt roads, also reflect strongly polarized light. These surfaces can attract mayflies and other aquatic insects, causing them to attempt to lay eggs on a car hood or a road surface rather than on actual water. The mismatch between the insect's evolved signal and the modern environment is a significant problem for the populations of many polar ataxis-dependent insects. The eyes of certain insects are also adapted to read the visual information encoded in the body patterns of other insects. Many bees and wasps have patterns on their bodies that include ultraviolet reflecting and ultraviolet absorbing regions. These patterns are thought to function as species recognition signals. When a bee approaches another bee, it can assess the ultraviolet pattern on the other bee's body to determine whether it is a member of its own species, a potential mate, or a potential rival. These visual signals are invisible to most vertebrate observers, which may be part of their function. They allow insects to communicate with each other while remaining inconspicuous to predators that cannot see ultraviolet. Fireflies demonstrate another dimension of insect visual communication. Fireflies produce their own light through bioluminescence. Different species of fireflies produce light in different patterns. The pattern is specific to each species. The male flies through the air, flashing its species-specific signal. The female, waiting on vegetation below, responds with her own flash if she sees a signal she recognizes. The timing between flashes is critical and species-specific. In some species, the flash interval is so specific that other species cannot decipher it. The visual system of the firefly must be tuned to detect these brief, species-specific flash patterns against the background of the dark environment. Research has shown that firefly eyes are sensitive to the yellow-green wavelengths, at which firefly bioluminescence is strongest. This specificity allows fireflies to communicate with each other while minimizing interference from other light sources. Different species of fireflies have evolved flash patterns that are as distinctive as Morse code sequences. The North American species Featureless, one of the most common fireflies in eastern North America, produces a characteristic J-shaped flash as the male dips during flight. The flash lasts roughly half a second. The female responds with a flash roughly 2 seconds later. If the timing is correct, the male lands nearby and the exchange continues. The entire communication system depends on precise visual timing. The firefly nervous system is tuned to measure these intervals accurately. Small deviations in timing are enough to prevent a response. This prevents cross-species mating, which would waste reproductive effort and produce no offspring. The visual system of social insects like ants is also worth exploring in detail. Many ant species live primarily in underground nests and move through the world guided by chemical signals. Their vision might seem less important in these circumstances. But ants that forage above ground need vision to navigate over long distances. Wood ants, for example, use visual landmarks to navigate through complex woodland environments. They memorize panoramic views of their surroundings and use these visual memories to guide them back to the nest after foraging trips. The compound eye of the wood ant is capable of forming a rough panoramic image. The ant stores this image as a visual snapshot and then attempts to move in a way that keeps its current view matching the stored snapshot. This navigation strategy is called snapshot matching. Desert ants of the genus Cattiglyphis live in open, nearly featureless environments where chemical trails would quickly evaporate. These ants are remarkable navigators. They can leave the nest, travel in a winding path over hundreds of meters while searching for food, and then return in a nearly straight line to the nest entrance. They do this using a combination of path integration, which keeps a running calculation of direction and distance travelled, and visual navigation using landmarks and sky polarization patterns. Their compound eyes are well adapted to the bright, high-contrast desert environment. The eyes are relatively large for the size of the ant, and they are positioned to give a broad visual field suited to landmark detection and sky sensing. Cattiglyphis ants have been studied extensively and have revealed much about the mechanisms of insect navigation. When an ant is experimentally displaced while returning to its nest, so that it finds itself in an unfamiliar location, it continues walking in the direction that its path integrator indicates should bring it to the nest. It arrives at a point that is displaced from the actual nest by exactly the distance it was moved. Then it begins searching in expanding circles. This demonstrates that the ant has a precise internal estimate of its position relative to the nest, based on accumulated step counting and direction measurement, and that it switches to a search pattern when this estimate tells it the nest should be nearby, but visual recognition of the nest entrance fails to confirm this. The use of visual learning in insects extends to individual recognition in some species. Paper wasps of the genus Polistes live in small colonies where the social hierarchy is important. Research has shown that these wasps can recognize the individual faces of members of their colony. They use visual patterns on the faces of other wasps to identify individuals and remember their social relationships. Wasps have compound eyes that normally produce low-resolution images, but the facial patterns of paper wasps appear to contain enough information for individual recognition within the resolution limits of the compound eye. This is a remarkable demonstration of how a visual system with apparently limited capability can still perform sophisticated recognition tasks, when the information it needs is structured in the right way. In an interesting experimental finding, wasps that had their individual facial markings made artificially uniform by painting them performed significantly worse. in social dominance competitions than wasps whose markings were left intact. This suggests that other wasps were actively using facial patterns to identify and remember individuals. Wasps with uniform, unrecognizable faces were treated as strangers by their nestmates even when they were not strangers. The visual recognition of individual faces, which we often think of as a distinctly primate or at least vertebrate ability, turns out to be present in insects with vastly smaller brains and much lower resolution visual systems. We should also consider the role of colour vision in mate choice among insects more broadly. Many species of butterflies show vivid wing patterns. These patterns serve several functions. They could be warning signals to predators, indicating that the butterfly contains toxic or unpleasant chemicals. They can be camouflage in certain environments, and they often function as mating signals. The visual system of butterflies is well suited to evaluating wing patterns. Because many butterflies can see ultraviolet light, their wing patterns often have ultraviolet components that are invisible to vertebrate observers. A butterfly wing that looks a solid blue or orange to a human observer may appear strikingly patterned to another butterfly of the same species, with bands or spots of ultraviolet reflecting scales, creating patterns that only another butterfly can fully appreciate. The great spangled fritillary butterfly, for example, has wings that look uniformly orange to a human observer, but under ultraviolet photography, clear patterning becomes visible. The ultraviolet pattern may encode information about the health and age of the individual. A younger, healthier butterfly can maintain better scale architecture, and thus produces a stronger and more defined ultraviolet pattern. The structure of insect eyes is also linked to the specific demands of hunting. Predatory insects have notably different eye arrangements than their prey. The praying mantis, as we noted, has forward-facing eyes that provide binocular overlap. This arrangement sacrifices the wide peripheral view that sideways-facing eyes would give. Instead, it provides precise depth perception in a narrow forward zone. This is the optimal arrangement for an ambush predator that needs to judge the exact distance to prey before striking. The mantis does not need to see behind itself. It is a sit-and-wait predator that does not need to run from anything. What it needs is precision, and its eye arrangement provides exactly that. Robber flies are fast aerial predators that catch other insects in flight. Their compound eyes are very large relative to their body size. In some species, the eyes are arranged so that there is a specialized acute zone pointing forward and slightly upward. This acute zone has a higher density of ommatidia than the rest of the eye. This means it has higher resolution in that specific direction. When a robber fly detects a potential prey item, it will typically position itself so that the prey falls within this acute zone. The fly then makes its high-speed interception flight. Combination of high temporal resolution, a forward acute zone, and a fast nervous system makes the robber fly a very effective aerial predator. Studies have shown that robber flies have a prey capture success rate of over 90%, which is extremely high for a predator. Dragonflies are among the most accomplished aerial hunters in the insect world. They catch prey in flight with a success rate estimated at around 80 to 95%. This remarkable performance depends heavily on their visual system. Dragonfly is a very large and nearly spherical, covering most of the head. They provide close to 360 degrees of coverage with a high-resolution zone directed forward and upward. Research has shown that dragonflies use a hunting strategy called motion camouflage. When pursuing prey, the dragonfly adjusts its own flight path continuously, so that it remains in a fixed position in the visual field of its prey. From the prey's perspective, the dragonfly appears to be stationary, even though it is actually approaching at speed. Only when the dragonfly is very close does it reveal itself by shifting position to make the final strike. This strategy exploits the fact that moving objects are easier to detect than stationary ones. By appearing stationary in the prey's visual field, the dragonfly avoids triggering the prey's motion detection alarm. The execution of this strategy requires sophisticated real-time visual processing. The dragonfly must constantly monitor the position of its prey, calculate the prey's predicted trajectory, and adjust its own flight path accordingly. All of this happens in the dragonfly's small nervous system, which contains roughly 1 million neurons. For comparison, the human brain contains roughly 86 billion neurons. With 1 million neurons, the dragonfly achieves a hunting performance that would challenge the most advanced artificial tracking systems. The efficiency of the neural processing involved is extraordinary. The dragonfly's neural circuits for prey tracking include a group of neurons known as small target motion detectors. These neurons are specifically tuned to respond to small objects moving against complex textured backgrounds. They do not respond to large-scale motion of the entire background, which happens when the dragonfly itself moves. They only respond to the motion of small objects, like other insects relative to the background. This selectivity is achieved through inhibitory connections from neurons that encode the background motion. The background motion signal is subtracted from the total motion signal, leaving only the residual, small, target motion. This is a form of visual computation that allows the dragonfly to track a tiny prey item even against a visually complex background of waving vegetation or a busy sky. The flow of visual information from the compound eye into the insect brain follows a complex but well-studied pathway. From the retinula cells in each oectidium, signals travel along nerve fibers into the optic lobe of the brain. The optic lobe is a large and highly organized structure that takes up a substantial fraction of the total brain volume in visually oriented insects. It is divided into several distinct layers of neural tissue. Each layer performs different kinds of processing on the visual signals. The first layer, called the lamina, performs initial processing, including a step that enhances contrast by comparing the response of neighboring cells. This process is called lateral inhibition. It works by having each lamina cell send inhibitory signals to its neighbors. A cell that is strongly activated suppresses the activity of cells around it. This means that a cell at the edge of a bright region, where the contrast is high, is less inhibited on its bright side than on its dark side. The result is that edges appear sharper and brighter than regions in the middle of a uniform area. This is similar to the lateral inhibition process that occurs in vertebrate retinas. Both systems use the same basic computational principle to enhance contrast and detect edges. The second layer, called the medulla, performs more complex processing, including the detection of motion direction and the separation of color information. Deeper layers, including the lobular and lobular plate, are involved in computing overall motion patterns and integrating visual information with other sensory inputs and motor commands. The lobular plate in insects such as flies contains large motion-sensitive neurons known as lobular plate tangential cells. These neurons respond to large-scale patterns of visual motion across the visual field. Different cells respond to different directions and speeds of motion. Some respond to forward motion, others respond to sideways motion. Others are tuned to the kind of rotational flow that is generated when the insect spins around its own axis. These cells have been studied extensively because they are large and easy to record from with the electrodes. They have revealed a great deal about how the insect brain extracts motion information from the visual world and uses it to control flight behavior. When a fly drifts off course in the wind, the visual world appears to rotate across its eyes. The lobular plate tangential cells detect this rotation and generate signals that drive compensatory movements of the head and body. This is called the optomotor response. It acts like a visual stabilizer, keeping the fly on course in the presence of disturbances. The same class of neurons is involved in the detection of approaching objects. When something approaches directly toward the fly, the image of that object expands rapidly on the retina. The expansion triggers specific neurons in the lobular plate that initiate the escape response. The fly takes off or jumps before the approaching object can reach it. The speed of this escape response has been measured precisely. From the time the visual stimulus begins expanding to the time the fly begins its escape movement, roughly 60 to 100 milliseconds pass. This is faster than most vertebrate reaction times. The pathway from visual input to motor output is short. It involves relatively few synaptic connections. Each additional synapse introduces a small delay. By minimizing the number of connections in the escape pathway, insects achieve response speeds that would be impossible in longer circuits. The visual system of bees has been studied particularly thoroughly because of the importance of bees as pollinators and as model organisms for studying learning and memory. Bees can learn to associate colours and patterns with rewards. When a bee visits a flower and receives nectar, it forms a memory of the flower's visual appearance. On subsequent foraging trips, it preferentially visits flowers that match this memory. This colour learning is remarkably fast. A bee can form a stable colour memory after as few as 1 or 2 successful visits to a flower. The memory is also long-lasting. A bee that has learned to associate a particular colour with food can retain that memory for days. Bee colour learning has revealed some interesting principles about how their visual system works. Bees learn colours most effectively when the colour is presented just before and during the reward. If the colour is shown after the reward, learning is less effective. This is the same temporal arrangement that is required for learning in many other nervous systems. It suggests that the bee brain uses a similar reward-based learning mechanism to the one found in much larger brains. In the bee brain, this mechanism involves the release of a chemical signal in the mushroom bodies, which are paired structures in the insect brain associated with learning and memory. The mushroom bodies receive both sensory information and reward signals. When a sensory stimulus is followed closely by a reward, the coincidence of these two inputs leads to a strengthening of the connection between the sensory representation and the behavioral output of flying toward that stimulus. Bees also learn to recognize patterns. They can distinguish between circles and stars, between stripes and spots, between symmetric and asymmetric patterns. However, bees do not appear to recognize shapes in the same way humans do. Humans identify shapes by their overall form and outline. Bees appear to use a different strategy. They analyze patterns based on their texture and frequency content. A shape with many small elements has high spatial frequency. A shape with few large elements has low spatial frequency. Bees are sensitive to these frequency properties of patterns. They use this information to distinguish between different floral patterns. Experiments have shown that when bees are trained on a pattern and then tested on modified versions of that pattern, they respond most strongly to modifications that preserve the spatial frequency content of the original rather than modifications that preserve the outline or overall shape. This is a fundamentally different way of recognizing visual patterns. The compound eye of insects is also connected to a phenomenon that reveals something important about the relationship between eye design and ecological niche. Many insects have compound eyes that are divided into distinct regions with different optical properties. In drone honeybees, the compound eye is divided into a dorsal region and a ventral region. The dorsal region, which faces upward, has larger facets than the ventral region. Larger facets collect more light and provide slightly higher resolution. The dorsal region of the drone's eye is used specifically to track the queen during the mating flight. The queen, like all honeybees, flies against the sky. The drone looking upward sees her silhouette against the bright background of the sky. The larger facets in the dorsal region provide sufficient sensitivity to track this small, fast-moving target. The ventral region, which sees the darker ground and vegetation below, has smaller facets that are sufficient for general orientations, but not for the precise tracking needed during the mating chase. The eyes of dragonfly larvae, which live underwater, are quite different from the eyes of adult dragonflies. The larvae have compound eyes, but they are smaller and less elaborate than adult eyes. The larvae hunt underwater, where the visual environment is different from the aerial environment. Light scatters and absorbs more strongly in water than in air. The range of useful vision is shorter. The larvae rely more on close-range detection of prey than on the long-range aerial tracking of adult dragonflies. As the larva transforms into an adult during metamorphosis, the entire visual system is rebuilt. The compound eye grows dramatically. New ommatidia are added. The optic lobes of the brain expand. The neural circuits for aerial vision are formed. This transformation takes place during a period of extraordinary reorganization of the entire body during metamorphosis. The visual perception of color in insects has also revealed something fundamental about how color is constructed. Color is not a property of objects. It is a perception that arises from the interaction between light, the photoreceptors in the eye, and the processing that occurs in the brain. Objects have spectral reflectance profiles. They reflect different amounts of light at different wavelengths. But the colour that an observer perceives depends entirely on which wavelengths that observers' photoreceptors are sensitive to, and how the brain combines and interprets the signals from those receptors. Two observers with different photoreceptor types will perceive the same object as different colours. This means that an insect looking at a garden of flowers perceives a completely different world of colours than a human looking at the same garden. A red poppy that is vivid and striking to human eyes may be nearly invisible against a dark background to a bee, which has no red-sensitive photoreceptors. A pale white daisy that looks simple and uniform to us may be a complex beacon of ultraviolet patterns to the bee. The garden that we see and the garden that the bee sees overlap in the same physical space. They contain the same objects, but they are, in terms of their visual appearance, completely different worlds. The idea that different organisms inhabit different sensory worlds was developed by the biologist Jakob von Wexkull in the early 20th century. He introduced the concept of the Umwelt, a German word meaning surrounding world or self-world. Von Wexkull argued that each animal lives within a sensory bubble that is defined by the capabilities of its sense organs and nervous system. The world it perceives is the only world it can act within. The dog's Umwelt is rich with odours that are invisible to humans. The bat's Umwelt includes a landscape of echoes. The bee's Umwelt includes a sky full of polarization patterns and flowers blazing with ultraviolet markings. These worlds do not compete. They coexist within the same physical universe, but they are as different as worlds can be. For insects, the visual Umwelt is shaped by millions of years of evolutionary pressure. Each aspect of insect vision that we have discussed is an adaptation. The wide-angle coverage of the compound eye evolved because predators approach from all directions. The high flicker fusion rate evolved because fast movement is both a threat to avoid and a resource to pursue. Ultraviolet vision evolved in step with the evolution of flowering plants. Polarization sensitivity evolved to allow navigation by skylight and detection of water surfaces. Motion camouflage evolved as a hunting strategy in a world where visual motion detection is a primary prey defense. Every feature of the insect visual system is the product of selection pressure, acting on genetic variation over immense spans of time. One of the more remarkable recent discoveries in the insect vision research concerns the way that some insects process visual information in the context of time of day. Bees have an internal clock that runs on a roughly 24-hour cycle. This circadian clock influences many aspects of bee behaviour, including the sensitivity of the visual system. At different times of day, the bee's visual system is tuned differently. The photoreceptors change their sensitivity. The neural circuits that process visual information alter their thresholds. These daily changes allow the bee to optimize its visual performance for the light conditions of different times of day. Early morning is dominated by shorter wavelengths. Late afternoon light is warmer and richer in longer wavelengths. The bee's visual system adjusts to these changes in a way that maintains colour constancy. Colour constancy is the ability to perceive the colour of an object as roughly the same under different lighting conditions. Without colour constancy, a flower that the bee has learned to recognise by its colour in the morning might appear to be a different colour in the afternoon, making recognition difficult. Colour constancy is a sophisticated computational achievement. It requires the visual system to estimate the overall colour of the illuminant and to subtract it from the perceived colour of objects. Even a relatively small nervous system like that of a bee can accomplish this. The mechanism used by the bee brain involves comparing the ratio of responses across different photoreceptor types, rather than using the absolute response of any single type. By looking at ratios, the system can compensate for overall changes in illumination intensity and colour. This is broadly similar to the mechanism used by the human visual cortex to achieve colour constancy, even though the bee brain is incomparably smaller. The compound eye also mediates a behaviour called optomotor response, in a way that has important practical applications. The optomotor response is the tendency of an insect to turn in the direction of visual motion in order to maintain a stable position relative to the world. It is essentially a visual tracking reflex. Insects navigating through environments with moving visual elements need to distinguish between motion caused by their own movement and motion caused by external events. The visual system uses the pattern of motion across the entire visual field to make this distinction. When the insect is turning, the entire visual field rotates in one direction. When the insect is flying straight, but a gust of wind moves vegetation, only part of the visual field shows motion. By analysing the pattern of motion across the full visual field, the insect brain can separate self-motion from world motion. Understanding how insects navigate and process visual information has inspired an entire field of engineering called bio-inspired robotics. Engineers and computer scientists have studied insect eyes and insect visual processing algorithms in detail. They've used this understanding to design cameras, navigation systems, and pattern recognition algorithms that mimic insect strategies. The wide-angle camera array, where many small cameras are arranged in a curved surface to cover a wide visual field, is directly inspired by the compound eye. Wide-angle miniature cameras of this kind are now used in surveillance systems, drone navigation, and medical imaging devices. Their ability to capture a wide field without moving parts is directly analogous to the function of the insect compound eye. The high sensitivity of insect motion detection systems has inspired algorithms used in video processing. Computer systems that need to detect moving objects in video streams use algorithms that compute the difference between successive image frames. This is essentially the same operation that insect visual systems perform when they detect motion by comparing the activation of ommatidiovus time. The lobular plate tangential cells of the fly have inspired the design of algorithms for computing optical flow, which is the pattern of apparent motion across an image field. Optical flow algorithms are widely used in autonomous vehicle navigation, drone stabilization, and computer vision research. The ability of bees to learn color associations and navigate to specific locations using visual landmarks has inspired machine learning approaches to autonomous navigation. Robots that use visual snapshot matching, directly inspired by the desert ant strategy, have been built and tested in outdoor environments. These robots can navigate back to a home base by storing a visual snapshot of the view from that location and moving in a direction that minimizes the difference between the current view and the stored snapshot. The approach is simple, computationally efficient, and surprisingly effective in complex real-world environments. The polarization-sensitive navigation ability of insects has inspired the development of polarimetric sensors for autonomous vehicles. Conventional GPS systems can fail in urban environments where satellite signals are blocked by tall buildings. A polarimetric sky compass, inspired by insect polarization navigation, can provide heading information based on the polarization pattern of the sky and does not depend on satellite signals. Prototype devices based on this principle have been built and tested. They represent a direct translation of an insect sensory capability into a practical human technology. The study of insect visual neuroscience has also contributed to our understanding of how neural circuits compute in general. The visual system of the fly has been used as a model system for studying neural computation for decades. The fly's visual system is accessible, well-mapped, and produces robust, reproducible responses to well-defined stimuli. The genetic tools available for working with fruit flies allow researchers to selectively activate or inactivate specific neurons and observe the effects on visual behavior. This has made the visual system one of the most thoroughly understood neural circuits in biology. The principles of motion detection, contrast enhancement, and directional selectivity that were first described in fly visual neurons have since been found, with variations, in the visual systems of many other animals including vertebrates. The horseshoe crab, which is not an insect but an ancient relative of spiders and scorpions, provided some of the earliest and most important research on how compound eyes work at the level of individual cells. The horseshoe crab eye contains large, easily identifiable receptor cells that can be studied with electrodes. Research on the horseshoe crab eye in the mid-20th century led to the first clear descriptions of lateral inhibition in a real nervous system. This work by Halden Keffer Hartlius and colleagues contributed to a Nobel Prize in Physiology or Medicine in 1967. The principles discovered in the horseshoe crab eye proved to apply broadly across animal visual systems. They explained many of the visual illusions that humans experience, including the Mach band illusion, in which we perceive dark and light bands at the edges of regions of uniform brightness. The brain's lateral inhibition mechanism is responsible for these perceptions. Insects, horseshoe crabs, and humans all share this fundamental mechanism, even though their eyes and nervous systems are profoundly different in other respects. Let us now consider how insects detect and respond to the visual world during their early life stages. Many insects go through complete metamorphosis. The caterpillar of a butterfly has a completely different body than the adult butterfly. The visual system of a caterpillar is very simple. It consists of a small number of simple eyes called stemmata. Stemmata are single-lens eyes. They have one lens focusing light onto a small number of receptor cells. A caterpillar may have 6 stemmata on each side of its head. These are capable only of detecting the general direction and intensity of light. They do not form detailed images. They tell the caterpillar whether it is in light or darkness, and roughly which direction light is coming from. This is sufficient for basic orientation and for finding leaves to eat. The caterpillar does not need high-resolution vision. It lives in a small world defined by the surface of vegetation. It uses chemical senses far more than visual ones to navigate this world. When the caterpillar pupates, everything changes. Inside the chrysalis, the caterpillar's body breaks down and is rebuilt. These stemmata are replaced by fully developed compound eyes. The small brain of the caterpillar is reorganized into the larger and more complex brain of the adult butterfly, complete with mushroom bodies for learning and large optic lobes for visual processing. When the butterfly emerges, it has a completely different visual system suited to an entirely different lifestyle. The butterfly needs to find flowers, identify mates, navigate over distances, and detect predators in the open air. All of this requires the wide-angle, colour-sensitive, motion-detecting compound eyes that it now possesses. The transformation of the visual system during metamorphosis is one of the most dramatic examples of developmental reorganization in biology. It involves not just the growth of new structures, but the selective death of old neurons, the migration of new neurons into place, the formation of thousands of new synaptic connections, and the pruning of connections that are no longer appropriate. All of this is orchestrated by a precise developmental program encoded in the insect's genes, modified by hormonal signals and by sensory experience during development. The same genes that specify the compound eye in the adult butterfly also specified the stemmata in the caterpillar, but under different developmental conditions they produce completely different structures. Grasshoppers and locusts do not go through complete metamorphosis. They undergo a more gradual process called incomplete metamorphosis. A young grasshopper, called a nymph, looks like a small version of the adult grasshopper. It has compound eyes from the beginning. The nymph's compound eyes grow with each molt, adding more ommatidia at each stage. By the time the grasshopper reaches adulthood, its eyes are fully developed. This more gradual development of the visual system means that young grasshoppers and adult grasshoppers use similar visual strategies, unlike caterpillars and butterflies, which have fundamentally different visual capabilities. One of the more striking aspects of insect visual biology is the role that eye position and eye shape play in signalling and communication between insects. The bold, widely spaced eyes of some insects are important visual signals to other insects and to predators. The large forward-facing eyes of certain robberflies and horseflies make them look formidable and may discourage smaller insects from attacking them. Some harmless insects have evolved markings on their bodies that resemble the large eyes of dangerous species, exploiting the visual discrimination abilities of predators. An eye spot on the wing of a butterfly or moth, when suddenly revealed to a predator that has approached closely, may mimic the appearance of a much larger animal's eye. The sudden appearance of a large eye-like stimulus triggers an avoidance response in many vertebrate predators. The predator hesitates or withdraws, giving the insect time to escape. The visual processing of eye spots by predators has been studied extensively with birds, which are major predators of insects. Birds have excellent colour vision and are highly responsive to the visual patterns of their prey. When a bird encounters a moth with its wings closed and its camouflage pattern invisible, it may begin to approach. If the moth then opens its wings suddenly, revealing large eye spots, the bird will often startle and back away. The eye spot pattern works because it taps into a deeply embedded fear response in the predator. Large eye-like patterns are associated with potential danger because large eyes belong to large animals that are potential threats. The moth's eye spots do not need to be a perfect representation of real eyes. They only need to be sufficiently similar to trigger the fear response. This demonstrates a remarkable point about visual communication. It is not only about transmitting information accurately, it is also about exploiting the visual processing systems of other animals. The moth does not know that its eye spots frighten birds. It simply has them because ancestors with eye spots survived more often than ancestors without them. The bird's fear response is also not a learned behaviour in most cases. It appears to be partly innate. The combination of these two evolved properties, the moth's pattern and the bird's response, creates a working communication system even though neither participant needs to understand what is happening. The visual abilities of flies have been particularly important in the study of visual neuroscience because of the availability of powerful genetic tools in fruit flies. The species Drosophila melanogaster, the common fruit fly, has been one of the most important model organisms in biology for over a century. Its genetics are exceptionally well understood. Every gene in its genome has been identified. Many genes have been studied in detail. Because the visual system of Drosophila is genetically tractable, researchers can create flies with mutations in specific genes involved in visual processing and observe the effects on the fly's visual behaviour. They can express light-sensitive proteins in specific neurons and use pulses of light to activate or silence those neurons selectively. They can label specific neurons with fluorescent markers and trace the complete path of a visual circuit through the brain. These tools have revealed the complete wiring diagram of the Drosophila visual system in extraordinary detail. The lamina, medulla, lobular and lobular plate have all been mapped at the level of individual cells and synaptic connections. Hundreds of distinct cell types have been identified and characterized. The computational role of many of these cell types is now understood. The direction-selective motion detection circuit, for example, has been traced in complete detail. It involves a series of well-defined cell types that collectively compute the direction of motion with high accuracy. This circuit uses a combination of excitatory and inhibitory connections time to produce responses that are large for motion in one direction than for motion in the opposite direction. The basic principle of this circuit turns out to be similar to motion detection circuits in the mammalian retina, despite the fact that insect and vertebrate eyes evolved independently and have entirely different structures at the cellular level. The convergence on similar computational solutions in very different nervous systems is a striking example of evolutionary convergence. As we slow down and approach the end of this quiet exploration, it is worth pausing on the scale of what we have been discussing. The insect compound eye is a structure that contains thousands of precise optical units. Each unit is a miniature optical instrument. Each one is surrounded by its own pigment cells that control optical isolation. Each one connects to a layer of neural cells that process its output. All of this is assembled during development from a single fertilized egg, guided by a genetic program that fits comfortably in a nucleus far too small to see with the naked eye. The precision of the construction is extraordinary. No human engineer has ever built anything with this level of miniaturization and functional precision at a comparable scale. And yet this structure, remarkable as it is only the beginning of insect vision. The eye collects photons. What happens to those photons after they're detected is equally remarkable. They're transformed into patterns of electrical signals. Those signals are processed through layers of neural circuits that compute contrast, motion, direction, color, and spatial pattern. The outputs of those computations are integrated with information from other senses and from the insect's internal state. The result is behavior. A bee finds a flower, a dragonfly catches a mosquito, a firefly finds a mate, a desert ant finds its way home across a featureless plain. All of this emerges from the interaction of light with a curved surface covered in thousands of tiny lenses and the neural machinery that lies behind them. The visual system of insects also interacts with their other senses in ways that are important for understanding behavior as a whole. The relationship between vision and the sense of smell in bees provides a clear example. When a bee approaches a flower, it begins to detect the flower's scent from a distance. The scent guides it toward the general location of the flower. As it gets closer, vision takes over. The bee uses the visual pattern of the flower to make its final approach and to select the correct landing position. Scent and vision together provide complementary information. Scent provides long-range guidance. Vision provides short-range precision. Experiments in which bees are trained with flowers that have scent but no visual pattern, or visual pattern but no scent, show that both cues independently contribute to learning. When both are present, learning is faster and more robust than with either cue alone. The bee brain combines information from both sensory modalities and forms a combined memory that is richer and more reliable than either memory would be alone. The sense of touch also interacts with vision in insects during close-range inspection of flowers. As a bee lands on a flower and begins to probe for nectar, it uses its antennae to feel the surface texture and temperature of the petals. Many flowers are slightly warmer at their center when nectar is produced. Bees can detect this warmth through their antennae. The combination of visual pattern, scent, and thermal signals all converge to guide the bee toward the nectar reward. The visual system initiates the approach. The other senses refine the final positioning. This multi-sensory integration is a fundamental feature of insect behavior and reflects the way that different sensory modalities evolved together as a coordinated system. The visual abilities of locusts have particular relevance because of the importance of locust swarms in agriculture. A locust swarm can contain hundreds of millions of individuals. Each locust must navigate through the swarm without colliding with its neighbours. The density of a locust swarm means that each locust is continuously surrounded by other flying bodies, each moving at speed in a slightly different direction. The visual system of the locust must be capable of tracking multiple nearby objects simultaneously and generating avoidance responses quickly enough to prevent collision. Research on locust visual neuroscience has identified neurons in the locust brain that respond specifically to approaching objects. These neurons, called lobular giant movement detectors and sending contralateral movement detectors, are specifically tuned to objects that are expanding in the visual field, which is the signal generated by an approaching object. They project directly to the motor system and can trigger rapid evasive maneuvers within tens of milliseconds. This system is important not only for collision avoidance within swarms, but also for escape from approaching predators. The visual acuity of insects varies not only between species but also across different regions of the same compound eye. This regionalization of visual acuity is a general principle of compound eye design. Most compound eyes have one or more regions of elevated acuity, where the ommatidia are more tightly packed together, or where the individual facets are larger. These high acuity regions are sometimes called acute zones or foveas, borrowing a term from vertebrate eye anatomy. The insect acute zone serves the same functional purpose as the vertebrate fovea. It provides the highest available resolution in the region of space where the insect needs the most visual detail. In predatory insects, the acute zone typically faces forward or upward toward the region of space where prey is most likely to be found. In insects that navigate by visual landmarks, the acute zone may be directed toward the horizon, where landmarks are most useful for navigation. The stalk-eyed fly is an insect whose compound eyes are placed at the ends of long, outwardly extending stalks. The eyes of this fly can be separated by a distance equal to or greater than the length of the entire body of the fly. This extraordinary eye placement has a significant effect on the visual capabilities of the fly. Widely separated eyes provide a larger baseline for binocular overlap, and therefore potentially better depth perception. But the primary function of the widely spaced eyes in stalk-eyed flies is almost certainly related to mate choice. Female stalk-eyed flies preferentially mate with males who have the widest eye spans. The eye span of the male is an indicator of his genetic quality. Males with wide eye spans are more vigorous and better fed than males with narrow eye spans. The female can assess the eye span of potential mates visually and choose accordingly. This has led to an evolutionary arms race in which male eye spans have become progressively more extreme over evolutionary time, driven by female preferences. The compound eyes of some insects are also sensitive to the direction of gravity through a mechanism involving not photoreception but proprioception. When an insect stands or hangs on a surface, the weight of the eyes pulls them downward relative to the head. Sensory cells in the eye socket detect this deflection. These cells tell the brain how the head is tilted relative to gravity. The brain uses this information to stabilize the visual image as the insect's head moves. In flying insects, the head is often decoupled from the body, meaning the head can rotate independently of the body to keep the visual world stable. The halters, which are small club-shaped structures in flies that evolve from hind wings, act as gyroscopes and provide information about the rotation of the body. The brain integrates the halter signals with the proprioceptive signals from the eye sockets to compute the correct orientation of the visual world. This allows the flea to maintain a stable visual image even during rapid, acrobatic flight maneuvers. The visual system of insects also plays a role in regulating the sleep-wake cycle. Many insects have a day-active phase and a night-resting phase. The timing of this cycle is regulated by the circadian clock, but the clock is entrained and reset each day by light signals detected by the compound eyes. The photoreceptors in the compound eye send signals about the overall level of illumination to structures in the brain that control the circadian clock. As daylight increases in the morning, these signals advance the clock and promote wakefulness. As light levels decrease in the evening, they promote the transition to rest. In addition to the compound eyes, there is a third type of eye found in some insects that is specifically involved in circadian entrainment. This is a light-sensitive structure in their brain itself, sometimes called the Hofbauer-Buchner islets in flies. These structures respond to light independently of compound eyes and contribute to the setting of the internal clock. The relationship between vision and flight control in insects is one of the most intensively studied topics in insect neuroscience. A flying insect must constantly monitor its own movement through visual feedback and use this information to correct deviations from its intended trajectory. This is a continuous process that operates many times per second during flight. The visual system provides three main types of information for flight control, and the first is translational optic flow, which is the pattern of the motion generated by the insect's forward movement through the environment. As the insect flies forward, the visual world flows past it from front to back. The speed of this flow depends on the insect's velocity and on its height above the ground. By monitoring the speed of translational optic flow, the insect can regulate its own flight speed and maintain a safe height above the ground. Bees approaching a landing surface slow down by monitoring the expansion of the visual image of the surface as they approach. When the image expansion rate reaches a certain level, they extend their legs and land. This approach control strategy does not require the bee to know its own speed or distance from the surface. It only requires that it monitor a visual variable that combines both pieces of information. The second type of visual information for flight control is rotational optic flow, which is generated when the insect turns or rotates. Any rotation of the insect causes the visual world to rotate in the opposite direction. By monitoring the rate of rotation of the visual world, the insect can detect unintended rotations caused by turbulence or gusts and generate corrective movements to stabilize its flight. The third type is binocular disparity, used mainly by insects with sufficient binocular overlap to estimate distances to objects in the forward direction. This is most relevant for landing and for the final stages of prey capture. The study of how flying insects use visual information for flight control has directly inspired the design of autonomous aerial vehicles. Small quadrotor drones use certain cameras and optic flow algorithms to navigate without GPS in indoor environments. They can follow corridors, avoid obstacles, and land on surfaces using exactly the same visual strategies that insects use. The algorithms were developed by studying insect flight behavior and then implementing the same computational principles in silicon. The resulting systems are simple, lightweight, and effective. They represent a direct application of biological insight to engineering. Before we leave the subject of insect vision entirely, it is worth considering one more remarkable phenomenon. Some insects can see through objects that are opaque to our eyes. This is not magic. It is a consequence of the different wavelengths that their eyes are sensitive to. Some plants have leaves that strongly absorb visible light but transmit near-infrared radiation. Near-infrared radiation has wavelengths just beyond the red end of the human visible spectrum. Some insects, particularly some species of wasp and butterfly, have photoreceptors that are sensitive to near-infrared wavelengths. To these insects, a leaf that appears solid and opaque to human eyes may be partially transparent, allowing them to detect objects on the other side. This capability could help plant-feeding insects detect the nutritional quality of leaves, as healthier leaves with more chlorophyll transmit near-infrared differently than stressed or damaged leaves. It could also help parasitic wasps locate host insect larvae that are concealed inside plant tissue. The sensitivity of some insect eyes extends well beyond near-infrared as well. Certain forest floor beetles that live in environments where visible light barely penetrates have compound eyes that are sensitive to the near-infrared thermal emission of warm objects. This is a completely different phenomenon from near-infrared reflection. Thermal near-infrared is emitted by objects based on their temperature, not reflected from external light sources. A warm mammal emits near-infrared thermal radiation even in complete darkness. Beetles sensitive to this radiation can potentially detect warm bodies in the dark. Research on this capacity is ongoing and not fully characterized, but the principle demonstrates that the electromagnetic sensitivity of insect eyes can extend into regions of the spectrum that most animals cannot access at all. The insect eye, in all its variations, is a window into the physical world that is both similar to and profoundly different from the window we look through. It evolved to solve the same fundamental problem that all eyes solve, extracting useful information about the environment from the flow of electromagnetic radiation. It solved this problem with different materials, different geometry, and different neural circuits than the eyes of vertebrates. And in solving it differently, it opened access to aspects of reality that are invisible to us. The ultraviolet markings of flowers, the polarization map of the sky, the thermal glow of warm bodies in the dark, the rapid flicker of motion at 300 frames per second. These are real features of the physical world. They exist regardless of whether any eye perceives them. But it took the evolution of the insect eye in its many remarkable forms to make them visible at all. The neurochemistry of the insect visual system is another area where insects and vertebrates show surprising parallels. In both insects and vertebrates, the initial step of vision involved a class of proteins called opsins. Opsins are light-sensitive proteins embedded in the membranes of photoreceptor cells. They bind to a small light-absorbing molecule called retinol, which is derived from vitamin A. When light strikes the retinol molecule, it changes shape. This shape change triggers a cascade of chemical reactions inside the photoreceptor cell that ultimately generates an electrical signal. The basic opsin-retinol system is remarkably conserved across the animal kingdom. The opsins of insects and the opsins of vertebrates are structurally similar. They work by the same general mechanism. This suggests that the molecular machinery for light detection evolved once, very early in the history of animal life, and has been adapted and modified in many different ways to produce the enormous diversity of visual systems we see today. The specific tuning of each opsin to a particular wavelength of light depends on the precise chemical environment around the retinol molecule. Small changes in the amino acid sequence of the opsin protein can shift the wavelength at which the opsin absorbs light most strongly. Over evolutionary time, gene duplication events have produced multiple copies of the opsin gene in many lineages. Each copy can then evolve independently, acquiring mutations that shift its absorption spectrum. This is how animals with multiple photoreceptor types evolved from ancestors with a single type. A duplication event produced 2 copies of the ancestral opsin gene. Mutations in each copy shifted the absorption spectra in different directions. Animals that inherited both copies could now compare the outputs of 2 differently tuned photoreceptors and thereby gain color vision. Subsequent duplications and divergences produced animals with 3, 4, 5 or more opsin types, each tuned to a different wavelength. The evolution of color vision is thus deeply tied to the evolution of the opsin gene family. In insects, this gene family has diversified extensively. Different insect lineages have gained and lost opsin types at different points in their evolutionary history. The ancestral insect probably had 3 opsin types, sensitive to ultraviolet, blue and green. Many modern insects retain this ancestral complement, but some lineages, like butterflies, have added additional opsins through further duplication and divergence. Others, like many beetles, have lost 1 or more of the ancestral types. The diversity of insect colour vision that we observe today is the result of this ongoing process of opsin gene gain and loss over hundreds of millions of years. The compound eyes of insects are not only organs of perception, they're also thermally important organs. Eye tissue consumes metabolic energy for the continuous biochemical processes involved in photoreception and signal processing. In many small insects, the compound eyes constitute a significant fraction of the total head mass. The metabolic activity of the eye tissue generates heat. In cool conditions, this heat can help maintain the temperature of the brain and optic lobes above the ambient temperature. This is relevant in the morning, when temperatures may be cool and insects need to warm up before they can fly efficiently. The warming of the eye tissue may assist in warming the adjacent neural tissue and help the insect become active more rapidly. Water beetles that dive below the surface face a different optical challenge. Water has a higher refractive index than air. This means that the focusing properties of a lens designed for use in air are different from those of a lens designed for use in water. Some diving beetles have compound eyes that are divided into a dorsal region and a ventral region by a horizontal band of modified ommatidia. The dorsal region of the eye operates above the water surface when the beetle floats at the surface. The ventral region operates below the surface when the beetle dives. The 2 regions may have somewhat different optical properties suited to their respective environments. Research on this remarkable eye design is still ongoing, but it illustrates the extraordinary range of adaptive modifications that the compound eye has undergone to suit different ecological demands. The migratory monarch butterfly uses its compound eyes as part of a sophisticated navigation system that allows it to travel from eastern North America to overwintering sites in Mexico each autumn. This migration covers thousands of kilometers and must be completed within a single butterfly's lifetime. The monarch uses the sun as a compass to maintain a southwesterly heading. It compensates for the changing position of the sun through the day using its internal circadian clock. The visual system of the monarch is tuned to detect the position of the sun and to integrate this information with the clock to compute the correct directional heading at any time of day. Experiments in which monarchs are kept in conditions that shift their internal clock by 6 hours show that they fly in a direction that is offset from the normal southwesterly direction by the amount predicted if the clock was shifted. This demonstrates that the clock really is being used to compensate for the sun's movement, and that the visual system provides the raw input of sun position to the clock compass navigation system. The compound eye of the monarch must be sensitive enough to detect the position of the sun, even when it is partially obscured by cloud. Research suggests that the monarch may also use sky polarization patterns, detected through the dorsal rim area of the compound eye, to determine the sun's position when the sun itself is not directly visible. The role of vision in insect communication extends beyond mating and species recognition. Some insects use visual signals to coordinate group behavior. Army ants are primarily guided by chemical trails, but visual signals also play a role in the behavior of the more visually capable species near the surface. Honeybees use a remarkable form of visual communication called the waggle dance to convey information about the location of food sources to nestmates. In the waggle dance, a forager bee returns to the hive and performs a figure-8 dance on the vertical surface of the honeycomb. The direction of the straight run in the center of the figure-8 indicates the direction of the food source relative to the sun. The duration of the waggle run indicates the distance. Bees watching the dance extract both pieces of information and use them to navigate to the food source. This form of symbolic communication, where a behavior represents a distant location, is one of the most sophisticated forms of animal communication known. The visual component of the waggle dance is the ability of bees to perceive and follow the movements of the dancer. The watching bees use the compound eyes to track the direction and speed of the waggle run. They must extract directional information from the dance movements and translate it into a flight direction relative to the sun. This requires the watching bees to understand the symbolic relationship between the direction of the dance and the direction of the food source. Whether this understanding is truly symbolic in the cognitive sense, or whether it is based on simpler stimulus response associations, is a question that researchers continue to debate. What is clear is that the visual processing required to follow and interpret a waggle dance is a sophisticated function of the bee visual system. The insect eye's vulnerability to damage and the mechanisms by which it protects and repairs itself are also worth noting. Strong ultraviolet radiation can damage the photoreceptor cells in the retina. Many insects have screening pigments in their eyes that filter out very short wavelength ultraviolet radiation while allowing longer wavelength ultraviolet to pass. These pigments are often yellow or orange in colour and are located in the accessory pigment cells that surround each ommatidium. They act as a sunscreen, protecting the sensitive rhodopsin molecules from the most energetic ultraviolet wavelengths, while still allowing the eye to detect the longer ultraviolet wavelengths that are useful for navigation and flower recognition. As insects age, the density and distribution of these screening pigments can change. Old bees, which have flown many thousands of foraging flights and been exposed to many hours of sunlight, show changes in their eye pigmentation that may reflect cumulative photodamage. The eyes of insects have also been the subject of intensive study in the context of biomimetic optical design. The corneal surface of insect eyes has a distinctive nanostructure called the moth eye pattern. This pattern consists of a regular array of tiny conical protrusions covering the corneal surface. The spacing between the protrusions is smaller than the wavelength of visible light. This nanostructure acts as an anti-reflection coating. It reduces the amount of light that is reflected off the corneal surface rather than entering the eye. A smooth glass surface reflects roughly 4% of incident light from each surface. The moth eye nanostructure reduces this reflection to less than 1%. This increases the amount of light that enters the eye and improves sensitivity, which is particularly important for nocturnal insects. The moth eye principle has been applied to the design of anti-reflective coatings for solar panels, camera lenses, and display screens. Anti-reflection films based on this design have been commercially produced and applied to a variety of optical surfaces. The study of insect vision has also illuminated fundamental questions about the relationship between eye design and brain size. Small insects with small brains have simpler visual systems than large insects with large brains. But the relationship is not simply proportional. Some insects with very small brains have evolved remarkably sophisticated visual processing capabilities for their size. The jewel wasp Ampulex compressa, which is a small parasitoid wasp, only about 2 centimeters long, is able to perform a precise neurosurgical procedure on cockroaches. It stings the cockroach in a specific location in the nervous system that temporarily disables escape behavior. It then leads the incapacitated cockroach to its burrow and lays an egg on it. The larva feeds on the living but immobilized cockroach. This behavior requires the wasp to locate cockroaches visually, orient precisely to deliver the sting, and navigate to and from its burrow. All of this is accomplished with a brain that contains only about 1 million neurons. The miniaturization of insect vision has reached extraordinary extremes in some parasitoid wasp species that are among the smallest known insects. Fireflies, which are tiny parasitoid wasps whose adults are less than half a millimeter in length, have compound eyes with only a few dozen facets each. Despite this extreme reduction, the flies are capable of finding host insect eggs, orienting to them, and depositing their own eggs inside them. Their tiny compound eyes, with their handful of ommatidia, are sufficient for the visual demands of their lifestyle. This demonstrates that even the most minimal compound eye design retains functional usefulness. The compound eye scales remarkably well from the tiny eyes of parasitoid wasps to the large elaborate eyes of dragonflies, always providing the same basic suite of capabilities in a form appropriate to the body size and ecological niche of the insect. Throughout this exploration, we have moved through the world of insect vision from the molecular level of opsin proteins to the behavioral level of dragonfly hunting strategies. We have seen that vision in insects is not a single thing, but a family of related solutions to a common problem. Each solution has its own strengths and limitations. Each is shaped by the specific demands of the insect's ecology and lifestyle. And each, when examined closely, reveals a level of elegance and precision that reflects hundreds of millions of years of evolutionary refinement. The ecology of insect vision extends into the night in ways that are worth exploring in more depth. Nocturnal insects face a fundamentally different visual challenge than their daytime counterparts. The amount of light available on a moonless night is roughly 1 billion times less than on a bright sunny day. Extracting reliable visual information from such dim conditions requires significant adaptations of both the eye and the brain. Beyond the superposition eye design we discussed earlier, nocturnal insects have evolved a range of strategies to improve their visual performance in dim light. Many nocturnal insects have larger facet diameters than related day-active species of similar body size. Larger facets collect more light. They do this at the cost of spatial resolution, because the angle between adjacent ommatidia is larger when the facets are larger. But in a dim environment, the priority shifts from resolution to sensitivity. A coarser but brighter image is more useful than a sharper but dimmer one. Neural summing is another strategy used by nocturnal insects to improve sensitivity. In this strategy, signals from neighbouring ommatidia are pooled together in the optic lobe. Pooling signals from neighbouring cells averages out random noise and produces a stronger, cleaner signal. The trade-off is again reduced spatial resolution, but in dim conditions, the improvement in signal quality more than compensates for the loss of detail. The visual systems of some nocturnal insects can dynamically adjust the degree of neural pooling depending on the light level. In bright conditions, the pooling is reduced and resolution is maximized. In dim conditions, pooling increases and sensitivity is prioritized. The temporal summation of light signals over time is a third mechanism. In bright conditions, the photoreceptors respond very quickly to changes in light. Their temporal integration window is short. In dim conditions, the integration window lengthens. The photoreceptor integrates light signals over a longer period before generating an output. This increases the total amount of light captured and improves sensitivity. The trade-off is reduced temporal resolution. Fast-moving objects become blurred because the slow integration window cannot keep up with rapid changes. But for a slowly walking nocturnal beetle searching for food on the forest floor, this trade-off is entirely acceptable. The beetle does not need to detect fast motion. He needs to see it all in near darkness. The combined effect of larger facets, neural pooling, and temporal summation means that some nocturnal insects can see by the light of a single candle at a distance of several meters, or navigate under a starlit sky that would appear completely dark to the unassisted human eye. The hawkmoth Dielaphila elpenor, which forages at flowers at night, has been shown to be capable of colour discrimination in light levels where humans are effectively colourblind. Humans lose colour vision at twilight because the cone cells become unable to respond at very low light levels, and only the rod cells, which are not colour selective, remain active. The hawkmoth retains colour vision at these light levels because its photoreceptors remain responsive, and its neural processing continues to compare signals across different colour channels even in near darkness. The hawkmoth's ability to see colour at night is directly relevant to its ecology. The flowers that the hawkmoth pollinates are often pale-coloured and open at night. They evolved in conjunction with the hawkmoth as a pollinator. The hawkmoth's ability to distinguish different flower colours at night allows it to learn and remember the visual appearance of rewarding flower species and to return to them efficiently on subsequent foraging flights. This is the same basic process of visual learning that we described in honeybees, but operating at light levels that are orders of magnitude lower. The fact that two species of insect with very different lifestyles both show visual learning and both use colour information suggests that visual learning is a deeply useful capability that has evolved independently in multiple insect lineages. The insect world contains one more visual wonder that deserves mention before we close. Some insects have evolved the ability to produce and detect circularly polarized light. Circularly polarized light is a form of light in which the plane of polarization rotates as the light propagates, forming a helical pattern. Circularly polarized light is very rare in nature. It is produced mainly by certain biological structures. The exoskeleton of the scarab beetle, for example, reflects left circularly polarized light due to the helical arrangement of chitin fibers in its cuticle. The iridescent green color of the scarab is partly the result of this structural arrangement. Some scarab species also have photoreceptors that are sensitive to circularly polarized light. This gives them access to a visual channel that is essentially private. Only other scarab beetles with the appropriate photoreceptors can perceive the circularly polarized signal. To any other observer, the beetle's surface appears simply green. The circularly polarized component is invisible without the right equipment, whether biological or technological. This private communication channel may serve several functions. It could allow scarab beetles to recognize members of their own species while remaining inconspicuous to predators. It could allow them to assess the health and quality of potential mates through a signal that is difficult to fake, because producing high-quality, circularly polarized reflection requires precise nanostructure, and it could allow them to communicate with other scarab beetles in close-range encounters without revealing this communication to outside observers. The study of circularly polarized vision in scarab beetles is relatively recent, and many details remain to be worked out, but it adds yet another dimension to the already extraordinary range of visual capabilities found in the insect world. As evening deepens into night and the garden outside grows quiet, somewhere in the leaves a moth is navigating by starlight. Somewhere a firefly is reading the flash patterns of potential mates. Saucera beetle is detecting a warm body by its near-infrared glow. And somewhere in the soil, in a dark underground chamber, a chrysalis is slowly completing the reorganization of a visual system, assembling thousands of en masse idea from the cells of a creature that only ever knew the simple perception of light and dark through 6 tiny stemmata. When the butterfly emerges, it will open its compound eyes and enter a world of colour, pattern, ultraviolet, and motion that its caterpillar self could not have imagined. That transformation, quiet and invisible in the dark, is perhaps the most remarkable visual event of all. We have travelled through many aspects of how insects see the world. We have considered the structure of the compound eye and the ommatidium. We have looked at the wide visual fields that compound eyes provide. We have thought about the high temporal resolution that allows insects to detect motion with extraordinary speed. We have explored the different colour channels that insects use, including their sensitivity to ultraviolet light. We've considered how the polarization of light serves as a navigational tool. We've followed the pathway of visual information from the retinular cells through the optic lobe and into the higher brain. We've looked at visual learning in bees, motion camouflage in dragonflies, facial recognition in wasps, the transformation of visual systems during metamorphosis, and the insect-inspired technologies that these discoveries have stimulated. Each of these topics opens onto deeper questions. The more we learn about insect vision, the more we appreciate the extraordinary diversity of solutions that evolution has found to the problem of seeing the world. A compound eye made of thousands of tiny units, each capturing a small slice of the visual field, is a radically different solution than the single-lens camera eye of a vertebrate, and yet it has proved at least as successful. The compound eye design has persisted for over 500 million years. It has been refined and adapted into thousands of variations. It works for the hunting dragonfly, for the foraging bee, for the navigating desert ant, and for the nocturnal moth. Its success tells us something important. There is no single right answer to the question of how to see. There are many answers, and the world is richer for having all of them. As you rest now and close your eyes, you carry in your mind a small map of how the world looks to creatures very different from yourself. The garden outside, if there is one, is a place of hidden ultraviolet markings. The sky above is crisscrossed with polarization patterns. The shadows that move across the ground are bright warnings to small eyes alert to motion. The flickering of artificial lights is a visible pulse to fast-sensing compound eyes. Insects live in this world. They've always lived in this world. They built their version of it long before we built ours, and they continue to navigate it quietly and successfully, guided by a kind of seeing that is in its own way as rich and as detailed as anything we know. Let your mind rest in that thought. The world is full of different ways of seeing, and each one is genuine, each one is real, each one is a window onto the same universe, opened at a different angle, letting in a different kind of light. Good night.